Om Swastiastu. Thank you for the opportunity given to us. We're from a small group discussion B9. We'll present our student project, NGS Based Technology on Medical Oncology. So here are the group members of B9 small group discussion. This presentation is divided into 3 chapters that are introduction, theory and discussion, and conclusion. Firstly, we'll start with chapter 1.
My name is Putri Cahyani with ID number 185. So I'll be presenting about the background of the paper. So every organism possesses a genome containing the biological information that is essential for living. In a human being, the genome is divided into 2 categories. The first category being nuclear genome, composed of a total length of 3 billion nucleotides organized into chromosomes. And then there's mitochondrial genome with a circular DNA molecule comprised of 16,000 nucleotides. In 1977, Sanger developed chain-terminating sequencing method to decrypt the whole human genome. However, it wasn't considered as an effective method to do sequencing, as it requires over a decade to read the whole human genome. It was in the year 1996 when this next generation sequencing was developed and was being used to parallel decipher the human genome within a short period of time, which is within a single day. It requires 3 main steps, which is library preparation, done by DNA fragmenting and conducting an in vitro adapter ligation, and there's amplification of the library occurs through either emulsion or bridge PCR to increase the availability of DNA, and then there's reading through the use of several cyclic RI sequencing methods, which consist of pyrosequencing, sequencing by ligation, and sequencing by synthesis. And then the next one that I'll be presenting is the problem formulations. The first being, what is the definition of next generation sequencing? And the second being, what are the methods used in next generation sequencing? How can next generation sequencing be applied into clinical oncology? And what are the advantages and challenges of next generation sequencing? That is for me. Thank you.
Hello everyone, my name is Ananta with the last 3-digit number of 198. Today I'm going to talk about the objective of this student project. The first objective is to know what is the definition of next generation sequencing. Next is to know what are the methods used in next generation sequencing. And next, to know how can next generation sequencing to be applied into clinical oncology. And the last one is to know what are the advantages and challenges of the next generation sequencing. I'm also going to talk about the benefits of this student project, which is improve infectious disease, diagnosis, inquiry, and tracking capabilities. And the second benefit is to develop of drugs that maximize therapeutic effects. The third is help guide patients to future clinical trials. And the fourth is help identify if someone has hereditary cancer. That's all from me. Thank you.
Hello everyone, my name is Anak, Agung, Rania, Kesuma, and the last 3 digits of my student ID number is 181. Next generation sequencing, or NGS, also termed as deep sequencing, is a massively sequencing technology that works in parallel, which offers high throughput, scalability, and speed. This new technology potentially enhances our ability to diagnose, interrogate, and track infectious disease by determining the order of nucleotides present in the entire genomes or targeted regions of RNA or DNA in a short time period at an affordable cost. According to the studies of S. Morganti et al., NGS platforms improve the process of DNA sequencing faster and simpler by employing microscopic, spatially separated templates of the DNA to massively parallelize the obtainable data. Today's highest throughput commercial instruments can generate nearly 1 terabase per run, allowing the sequencing of all coding exons of a genome and even a full genome in a short time and at a low cost. I think that's all I can explain about the definition of NGS. I'm sorry if there's any wrongdoings. Thank you.
Hello, my name is Nyoman Gede Wiraguna Paramartha, and my last three-digit number is 180. And I'm going to talk about NGS revolution for precision medicine. So NGS, or next-gen sequencing data, provides a more detailed analysis of antibody library multiplicity. NGS can also be used to study protein-protein interactions and antibody antigen binding. NGS has been successfully used to identify novel mutation in severe cancers, including bladder cancer, renal cell carcinoma, and many more. So that's it from me. Thank you.
First of all, thank you for the opportunity given to me. My name is Putu with last three digits of student ID 201. And now I'll present genomic NGS methods. NGS technologies have revolutionized genomic data studies by facilitating high-throughput sequencing of genetic materials derived from diverse sources. The first to use next-generation sequencing or NGS was implemented to genomic search to discover variants in DNA sequence. There are numerous methods are used, comprising whole genome sequencing, whole exome sequencing, and targeted sequencing. Whole genome sequencing, or WGS, is a methodology implemented to a whole genome where both coding and non-coding regions are sequenced. WES, or whole exome sequencing, is explicitly designed to only sequence coding DNA. And then targeted sequencing, specific regions of concern are isolated and sequenced. Many gene panels have been specifically designed for this purpose in order to focus time and resource on particular genes generally sequenced with a 500 until 1000 times coverage. And then lastly, we have basic steps on NGS data processing. There are basic calling, rate alignment, and variant identification or calling. So that's all my presentations about NGS methods with genomics. Thank you very much.
My name is Putu Iswari, Ardha Dharani, and my last ID number is 183. And now I will describe about transcriptomics. Transcriptomics is a technology used to study and understand the product of transcription from an organism, especially the sum of all its RNA transcript. All information contained in an organism will be recorded in its genomic DNA, which will then be expressed through the process of transcription. Transcriptomics has two contemporary techniques that are the main keys in field practice. The first one, microarrays, and the second one, RNA sequencing. Microarrays take measurements of a set of predetermined sequences, and RNA sequences capture all sequences using high-throughput sequencing. The existence of measurement of gene expression in an organism in different tissues, conditions, or time points help in knowing how a gene is regulated. It also reveals and shows details of organisms in the biological sciences. Transcriptomic techniques can also be helpful in inferring the function of previously annotated genes. Transcriptomics analysis has enabled study related to changes in gene expression processes in different organisms and help better understand disease in humans. Thank you.
Greetings, my name is Aryanda Maitri Anjasrini with 3 last digits of student ID number 188. Before we understanding what is epigenomics, we must first know some terms. The first one is genome. Genome is a complete unit consist of arrangement of DNA sequences and about 3 billion base pairs that makes an individual different from one another. The genome is being controlled and modified by an epigenome, which is the hereditary materials which is made of chemical compounds and protein. An epigenome that sticks to the DNA sequences of a genome could be said that it has marked the genome, and this mark could be passed by from generation to the next generation. The modification that occurring in a genome could be normal or abnormal. The normal one is the natural process of development and tissue differentiation, and the abnormal ones it could be response to environment exposure or pathogen that enters our body. The epigenomics learn that chemical changes that affect our body or our genes expression. And in epigenetic modification, it could be involved DNA methylation, histone modification, and altered DNA protein interaction. Thank you.
Hello, my name is Tjokorda Putri Mahadewi with my last 3-digit number is 189. So in here I will explain about the identification of novel cancer mutation. The first one is what is cancer? So cancer is heterogeneous disease that arise from the accumulation of DNA mutations and it can stop genes from working properly. And there's three main type control cell growth genes, which the first one is oncogenes, the second was tumor suppressor genes, and the last one is DNA repair genes. And there's a type of gene mutation. The first one is BRCA genes mutation. The second one is TP53 genes mutation, DNA mismatch repair genes, adenomatous polyposis coli genes, HER2, ERBB2 genes mutation, BCR, ABL fusion gene, KRAS gene mutations, EGFR gene mutation, ALK gene mutation, BRAF gene mutation, and TRK gene fusion, and the last one is driver mutation. I think that's all from me, and my friends will continue about it. Thank you.
Hello, doctor. My name is Michael Jonathan Raya with last three-digit names 182. In today's chance, I will explain about hereditary cancer syndrome and genetic testing. This is one of the application in the next generation sequencing, whether we can test if someone had a chance in developing hereditary cancer or not. So if a cancer is developed because of your lifestyle, your age, and environment, it is called sporadic cancer. There is no hereditary or family factors involved in it. But in hereditary cancer, there is a family factor that is inherited in someone's body, so they have a higher chance in developing cancer than a normal people. This happens because a mutation in a gene called tumor suppressor gene. This is a gene that codes certain function to prevent ourselves from developing cancer. If this gene is mutated, it will not do its function properly. So to test if someone has this mutation or not, we could do genetic testing to do initial screening and medical therapy to prevent us from developing further cancer development. And that's from me. Thank you.
Okay, let me introduce myself. My name is Amelia Hasikaranadewi with last three digits 207. Today I will explain about personalized cancer treatment. So, apart from identifying genetic and novel somatic mutation, another use of NGS is to improve rationally designed individualized medicine. To date, many studies have applied NGS for personalized treatment of cancer. So, this method is targeted to individual patients on the basis of genetic, biomarker, phenotypic, or psychosocial. It also explains that NGS is able to detect MIV less than 1%. Many methods like unique molecular identifiers or unique barcodes can help to increase the sensitivity and reduce the false negative. These methods are able to detect 59% of stage 1 or 2 of lung cancer patients with MIV around 0.1%. And have a good agreement between ctDNA response and radiographic response. Next will be explained by my friend.
My name is Gede Vivekananda Rusdi with student ID number 220251119. Today I'm going to present about the advantage of next generation sequencing. New technologies like nanopore sequencing present opportunities to perform analysis on DNA, RNA, or even proteins. The fourth generation sequencing has become known as new and very specific application field which depend on in situ sequencing in fixed cells and tissues and expected to provide major contribution in key areas. Every need in the genomic field can almost be fulfilled because of how the next generation system in RNA and DNA sequencing field is evolving. Currently, the second generation sequencing technologies respond to the most of the needs in the genomic field, while the third and fourth generation platform have the potential for a broader application spectrum with accurate and practical solution. With current NGS system or next generation system, sequencing system, it has become easier to obtain HD in much shorter times and could be in hours. Technologies like nanopore and single molecule real-time sequencing methodologies can be a solution for long reading problems and can be a solution in near future. For example, nanopore sequencing offers many solutions to the current problem of using MYGS for infectious disease diagnosis. It has been successfully utilized for pathogen detection, IMR prediction, and characterization of mixed microbial communities. That's all from me. Thank you.
Hello everyone, my name is Pratnyana Jayawardana with my last three digit student ID number 193. Today I will explain the challenges of NGS. Despite all the great benefits of NGS, the implementation of the technology in clinical laboratories has continued to grow slowly. The challenges of the implementation of NGS include the high initial installation costs, challenges in training due to the complex workflow, longer turnaround time compared to existing methods, and steep learning curve. That's it about the challenges of NGS. Thank you.
Hello everyone, my name is Ananta with the last three-digit number of 198. Clinical NGS is used to provide molecular rationale for appropriate targeted therapy, identify novel and rare cancer mutation, and detect familiar cancer mutation carriers. NGS provide a good solution for detecting rare variants. It's also important to recognize the current limitation of NGS analysis due to the limited number of prospect studies and sample sizes. Mutation screening has very limited negative predictive value. But over time, labs will increase driving forces. All of these trends will combine to make clinical cancer NGS testing less costly and complex to offer, thus making them more accessible to laboratories and patients. I will also tell about the suggestion of our student project. NGS technology is a new technology, and the knowledge that study NGS is still very narrow. At the high costs also make this NGS still difficult to develop. But over time, this rapid expansion has dramatically changed how we diagnose and screen for genetic condition, how we test, and how informative the results are. Learning about the recent advances in genetic testing and sequencing technologies has important implications for anyone working in healthcare and related sectors. This course offer a unique way for a professional to learn about genetic testing and its implications. We also hope that in the future NGS can help in the world of health. That's all for me. Thank you.
That's all our student project presentations. We are deeply apologize if there's any intentional or unintentional mistakes. Thank you for listening our presentations. Om Santi Santi Santi.